Hi, my name is Abhishek and I will be presenting our group's findings on how tau pathology in CSF mediates the impact of inflammaging on local sleep in older adults. Before we begin, I have no conflicts of interest to disclose. Alzheimer's disease is a public health crisis with no known cure. It is therefore imperative to understand the early biological and physiological markers of AD so as to facilitate the development of interventions that reduce risk in vulnerable populations. AD pathology is linked to sleep disruption at global and local scales, including decreased slow wave activity or SWA and fast spindle activity. Age-related central inflammation, also known as inflammaging, has been shown to impact and be impacted by AD pathology. Finally, inflammaging also shares a bidirectional association with sleep disruption at the global level, whereby poorer global sleep quality and duration increase inflammation, and vice versa. However, the relationship between inflammaging and local sleep is yet to be understood. This poses an important open question, the answer to which might inform interventions for AD. Therefore, in this study, our aim was to test whether inflammaging impacts local sleep, and if so, whether this relationship is mediated by tau pathology. In this study, 38 cognitively healthy older adults were recruited and underwent CSF collection through lumbar puncture at the University of Wisconsin-Madison in order to obtain fluid biomarkers, including phosphorylated tau, total tau, and YKL-40, which is a marker of glial activation. These fluid biomarkers were measured using ELISA assays. The subjects then participated in a high-density EEG sleep study using ASM sleep staging. These EEG data were filtered and cleaned of artifacts before generating spectral estimates for slow wave activity and fast spindle using multi-taper spectral analysis with 11 DPSS tapers. When we analyzed these results, we first ran multiple linear regressions between age and both YKL-40 and phosphorylated tau. Age showed significant effects on YKL-40 after adjusting for sex and AHI, but the impact of age on p-tau, although initially significant, did not show significance after accounting for covariates. Ultimately, the Sobel's test of mediation revealed a significant mediating role for inflammation in the age-tau relationship. Thereafter, we obtained mean absolute power in the SWA and fast spindle ranges and ran non-parametric correlations with a false discovery rate correction between this obtained power and total tau, phosphorylated tau, and YKL-40. SWA did not appear to be significantly correlated with any of them, but there were widespread and significant correlations between fast sigma power and all three biomarkers. Using a frontocentral derivation with peak significance across all three correlations, we ran a Sobel mediation model between phosphorylated tau, YKL-40, and fast sigma power. We found that phosphorylated tau had a significant mediating role in YKL-40's impact on local fast spindles, which remained significant even after adjusting for covariates such as age, sex, apnea-hypopnea index, and time between CSF collection and sleep study. We did run a similar model with total tau, however, this did not reach significance after accounting for all the covariates. Overall, these findings indirectly implicate inflammaging in local sleep deficits seen in older populations and set the stage for future work in several new and important directions. For instance, this study did not account for some important covariates such as years of education, ApoE genotype, and family history of AD. We will, however, be incorporating these into subsequent analyses. Yet another limitation of the study is in assessing how these relationships might interact with other biomarkers of inflammation, neuronal degeneration, and cognitive performance. Our future work looks at glial fibrillary acidic protein and soluble TREM2 for inflammation, neurofilament light protein and neurogranin for axonal and neuronal degeneration, and word pairs task performance for cognition. Finally, although the current study is cross-sectional in nature, our future work will be examining how these variables behave longitudinally. Finally, I would like to thank our entire team for all their contributions and hard work in this project, and I would also like to thank our funding body, the NIH. In particular, I want to acknowledge Dr. Mander for his help with this presentation. If you have any questions, please feel free to email me. My email is listed over here. Thank you so much.